Our next speaker is Judith, who is right here. And Judith was a PhD student with Gobind during 96 to 2001. And she is now at the Department of Structural Biology, University of Pittsburgh School of Medicine, Pittsburgh, among many other affiliations. And she's going to be talking about learning from Rhodopsin about Metabotropic Glutamate Metabotropic Glutamate Receptors. Have the abstract numbers, page numbers of 67 and 60. 66 and 67. So,
thank you very much. I'm going to call them mGluRs to avoid slipping over them. First of all, again, I would like to thank Gobind and the organizers for this wonderful occasion to be here. This is the outline of my talk. So I'm going to give a little bit of history, because the things that we do are essentially just continuations of hypotheses that Gobind has already brought forward. And especially I'm going to focus on allosteric modulation in the case of Metabotropic Glutamate Receptors, or mGluRs, and then summarize, and then I'm going to tell a story about Gobind too, but only at the end. So we may get the dancing after all. So one of the hypotheses that Gobind has brought forward is the structural coupling hypothesis, in which he proposes that the extracellular side and the transmembrane domain are structurally tightly coupled, and this is in particular because of this disulfide bond that locks them in place, and this is very important for rhodopsin stability and causes misfolding. And on the other side, the transmembrane domain is also structurally coupled to the cytoplasmic domain because the isomerization in the transmembrane domain changes the tertiary structure in the cytoplasmic domain. So this was really the driving hypothesis of my PhD thesis, and I worked on both of these sides. So the second hypothesis you have heard already in the last session. I think Wayne called it the central dogma. I call it the generality hypothesis, but it's the same thing. Basically, mechanism of receptor activation is conserved across all GPCRs. And so the fundamental idea being that you will have ligand changes induced changes in the protein structure that determine the functional state of the protein. And so this is just a slide illustrating what this really means in terms of diversity, because the G-protein coupled receptor family is the largest family on Earth. There are more than 7,000 GPCR sequences out there, and they're very diverse. They're grouped into three major groups, but with many orphan receptors as well, as we heard before. The largest group, the rhodopsin like family A receptors, but there's also the family B, the thyroid hormone receptor, and family C, metabotropic glutamate receptors. And so across these three major groups, the sequence identity is less than 10%. That is one of the major differences. But there's also a big difference in terms of their extracellular domains, because in both class B and class C GPCRs, you actually have quite large N-terminal additions to them. And this is extreme in the metabotropic glutamate receptor family, where you have around 500 amino acid additions, and the ligand in these two families actually binds in the extracellular domain, so not in the transmembrane domain, as it's predominantly in family A, although there are some exceptions here too. So in the case of the metabotropic glutamate receptors, there is signal transduction transfer from this extracellular domain, which, by the way, you can clip off, and it will still bind glutamate with the same affinity as in the full-length receptor. So the question of how the signal is transduced from the extracellular to the cytoplasmic site is an important and open question. But one of the interesting, relatively recent findings is that in this family of receptors, there aren't only this glutamate endogenous ligand, but people have also identified what is referred to as allosteric ligands. And it has been proposed that the allosteric ligand binding site is in the transmembrane domain. And there are two different types of ligands. There's the so-called negative allosteric ligands, which inhibit the response of the receptor to glutamate binding. And on the other hand, there's also positive allosteric modulators, which enhance the response to glutamate binding. But they both require that glutamate is there to activate the receptor, because with a few exceptions, neither allosteric type of modulator will activate the receptor by itself when the extracellular domain is present, and they can only serve as agonists or antagonists that actually you cleave off the extracellular part. And so they have become interested in this concept of allosteric modulation of G protein-coupled receptors, and it is not restricted only to the metabotropic glutamate receptors, but it's actually been found and experimentally demonstrated in all of the three major GPCR families. And in this table you can see a number of these examples. And if you look at where these ligands, the endogenous ligands for these receptors are binding, they can be either in the transmembrane or the extracellular domain. But for the allosteric ligand binding site, it has been proposed that, for example, in this case, when the endogenous ligand binding site is in the transmembrane domain, the allosteric ligand binding site is in the extracellular domain, and also vice versa. But recently it was also identified that there may be a cytoplasmic allosteric binding pocket, and I have I'll talk about it in a minute. I have a poster outside also with evidence from our lab supporting the presence of a cytoplasmic ligand binding site in Rhodopsin. So, given that ligands in different GPCRs can bind either in extracellular, transmembrane, or cytoplasmic domain, and in some of the receptors they can be endogenous, and other receptors they can be the allosteric ligand binding sites, I would like to propose that perhaps we can combine the two of Gobind's hypothesis in terms of generality as well as structural coupling, in that you may have ligands that bind in any one of these three domains, and they will be able to transmit information to either side of the membrane. And so this is the slide on the cytoplasmic allosteric ligands for the case of rhodopsin. So we have shown that anthocyanin compounds bind to rhodopsin. This is in a series of papers that just came out in photochemistry and photobiology. We also found that chlorophyll derivative chlorine A6 binds in the cytoplasmic domain, and all of these binding events actually modulate stability and functional properties of rhodopsin. And in the poster there's more detailed information on these structural and functional changes for the case of the chlorophyll derivative. There's also a recent paper where there was a screen carried out of a library of small molecules where a number of ligands have been identified that modulate G protein activation, and are also proposed based on docking experiments to bind in the cytoplasmic domain. So now moving to the metabotropic glutamate receptors, just a little bit of background why is it interesting to study these receptors. The number one reason is that glutamate is the most important excitatory neurotransmitter in the brain, and is therefore involved in learning, memory, and anything you can think of in terms of brain actions. And thus, not surprisingly, these receptors are involved in a number of different diseases ranging from schizophrenia, addiction, pain, depression, migraine, etc. The role of metabotropic glutamate receptors is not in direct glutamate transmission because that is accomplished by the ionotropic glutamate receptors, but instead these, which are ion channels, instead the metabotropic glutamate receptors play modulatory roles, and so they will change the response of neurons to glutamate. And so that's why they are believed to be potentially useful drug candidates. So the extension of the generality hypothesis for the metabotropic glutamate receptors is spelled out in this slide. So there we have Rhodopsin with its transmembrane retinal ligand, and the metabotropic glutamate receptor where the endogenous ligand is outside, but there's a proposed allosteric binding site in the transmembrane domain. And so we asked the question of whether these ligands here would bind structurally and perhaps functionally in a similar way like retinal does in rhodopsin. And so the approach that we have taken is a computational approach. We use the dark state crystal structure, Kolchevsky structure we use for this, generated an alignment of the transmembrane regions for the glutamate receptors. I didn't mention there are 8 different types of glutamate receptors, so we created models for all 8 of them because they have different ligand specificities which we are going to exploit. And so we collected all of the different allosteric ligands that have been identified and docked them using 2 different docking softwares, and then selected the best model based on energy and buried surface. So I should like to point out that these are completely unbiased docking experiments where we place a grid around the entire protein and find where would the ligand bind with the highest energy and most buried surface. And then we take the dot models and analyze the binding pocket and look at whether the critical residues in proximity to the ligand match with published experimental results. And so what you can see here is the binding pocket for, I think this is MPEP, I forgot now. So one of the, I think this is mGluR5 bound with MPEP, and this is the Rhodopsin structure with 11-cis retinal. And you can see just by comparing the models that the ligand binding pockets are roughly in similar domains, supporting the idea that this could be a conserved binding pocket. And we have a little bit of experimental results supporting that there may be some conservation in the binding pocket, in that this particular ligand here, 3,3'-difluorobenzaldazine, which is an mGluR5 modulator, actually appears to be able to bind to rhodopsin as well. So this is a very preliminary experiment, but what we did is bleach rhodopsin. You can see here the absorbance spectrum and here the difference spectra. So we bleached rhodopsin and then regenerated with 11-cis retinal in the presence and absence of different concentrations of this ligand. And you can see that in increasing amounts of the ligand present, there's a decreasing amount of regeneration of rhodopsin, suggesting that maybe the DFB is actually competing with the retinal. So this supports the idea that the binding sites are in different, in similar structures, but does this mean also that they would have similar functions for these proteins? So in the case of rhodopsin, we have 11-cis retinal in the inactive state, and then a conformational change for the all-trans beta-2, all-trans-bound beta-2 state. And so the hypothesis would be now that functionally this inactive rhodopsin state should correspond to metabotropic glutamate receptor bound with a negative modulator, while the beta-2 state should be mGluR bound with a positive modulator. And so in order to test this hypothesis, we need structures for these receptors for active and inactive models, and so the problem is that we only have a crystal structure of the inactive state. And only recently, as you know, there is an opsin structure that may have some resemblance to the active state, but we don't have an actual structure of the meta-2 state. And so what we did is use a model that we generated in collaboration with Yvette Bahar. This is based on a technique called anisotropic network modeling, where you take a protein, so this is schematically here shown for some random protein on the left-hand side, where the amino acids are just represented by single points for each amino acid. So this is a coarse-grained model with not considering any of the atomistic details. And so we now replace the actual covalent bonds connecting the residues with a so-called virtual bond representation, where we now consider linkages between amino acids occurring if they are within a certain cutoff distance. And you can then use this type of structural analysis to create a so-called Kirchhoff matrix, in which you look at how many neighboring partners each has, and each of these bonds is represented by a harmonic oscillator, and so this allows you to calculate what types of conformational changes are amenable to these residues within that context of the structure. And so you can then make predictions on what would be the conformational changes that are inherently accessible to a particular protein structure. And so this movie here shows a switching between the inactive dark state and our predicted activated model. And so we have compared this model with the data that you heard summarized in the previous session. And so it has this feature of helix 6 moving out, but we actually predict also that other helices are moving too, but they're moving conservatively, so the relative distances to each other are not changing that much. So that's still something that I think is also consistent with the experimental data, that there might still be these changes. And so we then use this model to now repeat the same protocol that I showed you before, but we are now not just docking the ligands to the homology models based on the inactive state of rhodopsin, but we also use our light-activated model and create homology models, and now dock these ligands to both of these types of structures. This slide here shows you all the ligands that we collected with the functional information, whether they are negative or positive modulators, for which subtype they are supposed to be specific. These are the structures of the ligands. And you can see here, for example, one of these ligands, MPEP, docked to its mGluR homology model, and so you can see they bind again in this pocket, similar to the retinal pocket, but you can see that the orientations are different, and so you would expect them to have different neighbors and different binding entities. And so we validated the models with available published experimental data. Then there are two fairly extensive papers, one on mGluR5 binding to 3'-DFB, which is a positive modulator for mGluR5, and another paper on MPP binding to mGluR5, and it's a negative modulator. These are the two structures. And then we compared what are the 5 angstrom residues in the binding pocket. And so you can see here, this is the reproduction of the experimental data, which says that these are the residues that are important for binding of the ligand. And you can see that in the structural model in green are those residues that we also identify within the 5 angstrom binding pocket in our models. And you can see also that there are differences between the active and the inactive models. So this data fits quite well with the model that's validating this, and the same is true for the DFB results. You can see here again the experimental data and the correspondence in our models, and we also predict additional residues, which may be still important. So we have some confidence in that these models provide some coarse-grained useful information about ligand binding and metabotropic glutamate receptors. And so we went ahead then and docked all of these ligands to their respective specific receptors. And what you can see here is a difference in energy between the ligands docked to the active model of that particular subtype versus the inactive model. And shown in green are the positive modulators, and red the negative modulators, and in yellow the neutral modulators. And so we use these two different types of software, and what you can see is that what we would expect is that the green ones should have negative values and the red ones should have positive values when we subtract the inactive models from the active model. You can see that with the exceptions of some outliers, this seems to be largely true for all of these different ligands with these different types of receptors. So this really supports the hypothesis that the ligand binding pocket in part is conserved between rhodopsin binding to retinal and metabotropic glutamate and receptors binding to these allosteric ligands, and that they also have the same functional consequences in terms of preferring active versus inactive structures. So in summary, I talked about Gobind's hypothesis, which are really the starting points for us, and the extension to our understanding of allosteric modulation in GPCRs, and we think that that there can be binding sites throughout GPCRs that can be amenable to each of them, regardless of where the actual endogenous binding site is, whether extracellular or transmembrane. This is something that I think may have importance in stabilizing receptors, modulating their activity, etc. Then I talked about metabotropic glutamate receptor analogy to rhodopsin. We identified a conserved structural site that highly overlaps with the retinal, and we have preliminary experimental evidence with rhodopsin that it can actually bind in the ligand binding, in the transmembrane ligand binding pockets, an mGluR allosteric modulator. And on the other side, in terms of mGluRs, we predict that the active and inactive models resemble the all trans and 11 cis-retinal bound states of rhodopsin. So, future work, we actually think that this might be something useful for the GPCR family in general. Perhaps we can predict for other GPCRs, give me a ligand and I tell you whether it's going to be an agonist or an antagonist. And we are currently in the process of extending these studies to better autonomic receptors because there's so much data available. But we are also looking at this from the other side in terms of experimentally validating these allosteric modulation hypotheses with one of the subtypes, in particular subtype 6. So I would like to deeply acknowledge Gobind for everything that he taught me, as well as the Corona Lab. The project that I described on metabotropic glutamate receptor is actually a small part of what we are working on. We also work with rhodopsin and other projects in computational biology. But the two students that have done all the work that I described today is Navina Yanamala, who just graduated, and Kalyan Chirupura, who is working on the mGluR6 expression in biophysical studies. And Yvette Bahar is our collaborator in predicting conformational changes, and Barsha Kising, who is also now graduated, was the student that predicted the structures. And the work is funded by NSF, NIH, and the Humboldt Foundation. So do I still have time? So this is my story. So every Saturday morning, Gobind used to bring this box with donuts to the lab. So we all thought it was very, very, very nice. But maybe you noticed that actually there is one donut of each kind in this box. And originally we thought it's just diversity, but it turns out that there is a reason for this. And slowly we figured out that in Gobind's mind, this box doesn't look like this. but it looks like this. So by the end of the day on Saturday, you would know who had been there.